Thank you. Our next speaker is Roy Haast from Aix-Marseille University, and the title is Towards Topography-Driven Stereotactic Targeting of the Zona Incerta.
Hello, everyone. It's a great pleasure to present to you today. I do this also on behalf of all my co-authors, and this project is led by Jonathan Lau. So I will be talking about the zona incerta, and especially how we can improve the targeting with deep brain stimulation using brain images, MRI images. So next slide. I have nothing to disclose. So the zona incerta, the name is quite mysterious, and then initially was also described by August Forel as an immensely confusing area about which nothing could be said. So it's a very famous quote, and here you can see the original cover page from the original manuscript and the original drawing, where you can see that the zona incerta is quite close to the thalamus and also close to the red nucleus. Luckily, we can say more things now about the zona incerta. You can see a little bit of the history of what is known about the zona incerta in terms of function. And you can see there's a change in the different focuses before and after the year 2000. So before 2000, it was really focusing on drinking and feeding behavior, and after that, it really got more known of its role related to deep brain stimulation. You have the dark blue versus the orange color. So when we dig a little bit in the structure and function, we can roughly subdivide the zona incerta in two different parts. Going from more rostral to caudal, we have the rostral zona incerta, the dorsal, ventral, and the caudal zona incerta. These have been linked to fear and anxiety processing, arousal, attention, and motor control. And especially the, can I go back? Yeah, this one. No, this button. So, like I said, there has been more and more attention related to the deep brain stimulation and its effectiveness to treat certain disorders. And especially the caudal zona incerta has been, stimulating this caudal zona incerta has been quite efficient to treat Parkinsonian tremor as well as essential tremor. While the rostral zona incerta has been linked more to, and treating with DBS to treating symptoms related to obsessive-compulsive disorder, or OCD. So here you can see an example of a study that was looking into the Parkinsonian tremor and the effectiveness of stimulating the caudal zona incerta. You can clearly see the effectiveness of reducing several symptoms, showing quite good results. However, currently, targeting of the caudal zona incerta is done quite indirectly using coordinates relative to surrounding structures like the subthalamic nucleus, as well as the red nucleus. And you can see examples, two examples shown here on the left, how this is done. We can imagine that can be not the most efficient way, not the most effective way to improve the outcome robustly across a lot of participants. So initial work by our lab, we tried to improve this a little bit by using quantitative T1 measures obtained at 70 MRI. So we obtained 70 MRI T1 data across 31 subjects to calculate a T1 template, and we used this T1 template to segment the zona incerta as well as the surrounding structures to characterize the T1 profile, so to obtain more individualized targets based on the T1 map. So you can see an example of how the initial targeting method, indirect targeting method, is overlaying with our segmentation label. So you can see it's actually at the border between the zona incerta label and FCT map, so the thalamic tract. So this was our first exploration of the zona incerta using 70 MRI data. Now we wanted to go more in detail and see if we can further refine our targeting strategy. So for this, we dig into the connectivity between the cortex and the zona incerta. So we know from non-human primate studies there's actually a gradient of connectivity between the zona incerta and the cortex. Going from the more rostral part to the caudal part, you can see that the caudal part of the zona incerta, which we know is linked to locomotion and motor control, is also more connected to the primary motor cortex. While the caudal, or that the rostral part is, which was linked to fear anxiety processing, is more linked to the prefrontal cortex and the frontal part of the brain. So this was done on non-human primates. So can we also extract the same kind of information in humans? And can we also use this information then to guide or refine our DBS targeting strategies? So this was the objective of our study. So for this, we used and leveraged this original segmentation labels from our initial study, as well as the high-resolution, high-quality diffusion data provided by the 70 Human Connectome Project data. So we use this to run probabilistic topography between the zona incerta seed label and the cortical regions defined by the Human Connectome Project multimodal atlas. This gave us the connectivity matrix for all our subjects, quantifying the number of streamlines between our seed voxels as well as the cortical regions. And when we then concatenate and average across all the subjects, the number of streamlines looks like this. So you can see that the majority of the streamlines are connecting to the motor regions, as well as a little bit to the medial and anterior part of the brain. So we then quantified the similarity between each voxel in terms of its connectivity to the rest of the brain. This is what's called an affinity matrix. And then we summarize this in two different ways. So we use diffusion map embedding as well as spectral clustering. I have no time to go really into the methodological details, but in the basics, the spectral clustering will provide you the more classic discrete clusters, while the diffusion map embedding will provide you more transient, continuous transitions of connectivity differences within our structure of interest. So here you can see the result when we perform the spectral clustering. So you can see the clusters are arranged more rostrally, organized more along this rostral to choroidal axis. So they run from blue to red, orange, green, brown, and pink along this axis. You can see how it's then connecting to the rest of the brain, which is done in a similar fashion. It's the same result, different representation. So on the cortical surface, we took a winner-took-all approach to quantify which parts of the brain are more connected to which clusters. And then here you can see the result when we look more at the diffusion map embedding, use the diffusion map embedding and the cortical gradients that are extracted from this. So we have the two primary and the secondary gradient. And it's actually showing the same kind of organization where we see a transition of connectivity from more rostrally to caudally, where here the blue part is more connected to the non-motor regions, especially the frontal part of the brain, and while the red part is more connected to the motor regions of the brain. A second gradient shows kind of the same organization, but it's emphasizing more the differences between the middle zona incerta, connecting more the premotor and the prefrontal cortex versus the rest of the brain. So then we leverage these two gradients to formalize a 2D gradient space where we plot the gradient 1 value on the x-axis, and the y-axis we plotted the gradient 2 value. So this allows you to visualize the two gradients in one plot, and then also to allow you to overlay the cluster solution, and which shows you the nice separation of the clusters based on these 2D gradients representation. So then the next step what we have to do is how can we leverage these gradients and these cluster solutions to fine-tune our DBS targets, zona incerta targeting. So we took one example patient which had 100% improvement after DBS, targeting the caudal zona incerta. You can see in here a reconstruction of the electrodes in the specific patients and how they were placed within the zona incerta with respect to the different clusters. Here you can see the same representation of the gradients as well as the clusters overlaid on this representation. And then the most important thing that I want to highlight is that you should look at these black markers which represent the VTA, so the activated voxels with the DBS. You can see how they are positioned with respect to the two gradients. So for both the left and the right hemisphere, you can see for the left hemisphere it's really overlapping with the green cluster. For the right hemisphere, it's more overlapping with both the brown and the green cluster. So this is a more quantification. So and again, it's showing the same as what I just said. So for the left and right, it's more the green cluster, cluster 4, while for the right, it's overlapping with both cluster 4 and 5. So this is an NS1, so this doesn't really tell you, okay, what is the best coordinate. So, but it gives you an insight of how we can use it. So the next step, what we wanted to do is to explore this at a larger scale. So we leveraged some data that was put online, and we were very happy with this by this paper from Barbara Hollander from the Andreas Hohn group, which was published last year in Nature Neuroscience. But this is focusing more on the subthalamic nucleus, but it provided the E-fields, so the stimulated voxels, for a lot of subjects that we could leverage and replicate our analysis. So I want to focus on two different example cohorts, so the Parkinson's disease, which has 94 subjects, as well as the OCD group with 90 subjects. So we did repeat the same kind of analysis, so we constructed the gradients as well as the cluster using this 2D gradient representation. And then we computed heat maps of which coordinates based on these gradients were mostly targeted for the two cohorts. You can see there's differences between the two cohorts, where the PD is more targeting the orange-blue clusters, so the more bright colors on the heat map, while in the OCD patients, the SDN is more targeting what color is this? Yeah, green and pink clusters. So this is just the number of patients. So what we also wanted to see, okay, which coordinates lead to the strongest improvement. So that's what we see here. Here you can see that it's similar to what we saw before, that, okay, so it's the more orange, brown, red clusters that shows the best improvement, like up to 80% improvement with the DBS on versus DBS off. But for the OCD, it's also more on the other side of the spectrum in this 2D gradient space. So this is a proof of concept. We are on our way to explore this further, but yes, we can use diffusion and right to resolve the topographic organization of the ZI. We can replicate the rostro-color gradient that was observed in non-human primates. And this gradient cluster-based representation might assist with the fine-tuning of the DBS targets based on patient-specific topography. We need further validation for the ZI, but the results in SDN look very promising. I want to thank all the co-authors. I also want to refer you to another poster related to zona incerta by Violet, and I'm happy to also answer any questions now or later at my poster tomorrow. I want to thank you for your attention. Thank you.
Thank you. So we have a question from the audience. Your work demonstrates a rostrocaudal cortico-insertia gradient and suggests cluster and gradient-based coordinates that you can personalize for targeting. Have you considered integrating individual patient functional connectivity profiles with resting state fMRI with diffusion-derived gradients to further refine stimulation targets and improve clinical outcomes?
So that was a long question. I'm not sure if I followed everything, but so if I considered functional connectivity, yes, we definitely considered. We have not really dug into these results, but it's something that is worth, of course, exploring and see if this is showing the same kind of direction as what we saw with the structural connectivity. I forgot the other part of the question. That was it? Okay.
Thank you so much.
Okay, thank you.